Throughout history, the ability to fly has been a coveted trait in various contexts. From the Wright brothers to Superman, everybody wants to fly, a desire that has driven many groundbreaking inventions such as airplanes, drones, and helicopters. This desire to fly leads us to question why birds, that belong to a clade that can traditionally fly, would lose that ability over the span of their evolutionary history. Why is it that large birds can't fly compared to their other feathered relatives? While some island birds evolved with flightlessness due to the absence of large predators, flightlessness within ratites, a term recognized for large flightless birds, is a trait that has evolved separately due to convergent evolution.
There are two living clades of birds, the Paleognathae and the Neognathae. Paleognathae consists of the tinamous and the ratites. Tinamous are volant, meaning they have the ability to fly, whereas the ratites are a flightless group that includes ostriches, rheas, emus, cassowaries, and kiwis. The evolution of flightlessness in ratites has been the subject of great debate for many years. One of the most compelling hypotheses suggested that the breakup of the supercontinent Gondwana fragmented a population of flightless ratite ancestors that evolved in place, creating today's ratites. But where do the volant tinamous fit in this tree? While it was previously thought that Paleognathae was a monophyletic group with two primary lineages, the tinamous and the ratites, molecular phylogenetic studies have found that tinamous aren't distant cousins of ratites at all, but are embedded within the group. This suggests a paraphyletic relationship, where ratites and tinamous evolved from a volant ancestor, and flightlessness was evolved several times in ratites.
Cynthia Foe and Daniel Field from Yale focused their research on how exactly two ratites, emus, and ostriches lost their ability to fly in their journal article titled Distinct Developmental Pathways Underlie Independent Losses of Flight in Ratites, published in July 2017. These scientists set up an experiment to test their hypothesis that flight was lost between the ostrich and emu independently through convergent evolution. To test this hypothesis, Foe and Field used eggs from 4 species, ostriches, emus, tinamous, and chickens, to analyze embryo development and determine the ultimate explanation for this phenomenon. The scientists incubated the eggs in optimal temperature and humidity for each species of egg that would be ideal for hatching. For the ostrich, chicken, and tinamous, they used a candling technique that used backlight to show embryonic details through the shell. An egg tapping technique was used for the emus since their eggs are too opaque for effective candling. During the incubation period, the researchers completed a staining process to monitor embryo development along with morphological analysis. Blue dye was used to represent cartilage, while red dye was used to represent calcified bone.
After this incubation period, the embryos that were free of genetic abnormalities were analyzed on 3 different criteria: body, wing, and leg length, which can be seen here in Foe and Field's Figure 2a. In regards to body length, the average growth rate was found to be highest in the ostrich embryos, followed by the tinamous, emu, and chicken embryos, respectively. As can be seen in Figure 1b, with the x-axis representing incubation time and the y-axis representing wing length, the average wing growth rate in terms of percent of incubation was found to be highest in the ostrich embryos, followed by similar rates between the tinamous and chicken, and then slowest for the emu. Additionally, researchers noticed a delayed onset of growth in the ratites, and that the ostrich embryo grows faster and longer than the other species, which results in a larger chick being born. Since this diagram, along with the previous information, aid in showing higher growth rates in both the body and wing of ostriches, the researchers were able to conclude that they go through hypermorphosis, which is the excessive growth of a body part, as well as paramorphosis, which is the maturation of a species past adulthood. On the contrary, the slow growth rates of the emu suggest patomorphosis, or the retention of juvenile traits. This difference in developmental time is termed heterochrony and accounts for the differences in size between the species as well as their inability to fly. Foe and Field found that the wing length and leg length were negatively associated with body size, which meant that the smaller the legs or smaller the wings, the larger the body size of the species.
Flight loss within the Paleognathae clade is a prime example of convergent evolution and paraphyly based on the embryonic and phylogenetic analysis. Recognizing that flightlessness is not a monophyletic trait within the clade allows us to see more patterns within the embryonic characteristics of the group and more effectively gauge how flightlessness evolved independently. Previous genomic evidence was showing more of a monophyletic relationship with ratites. However, the difference between the wing growth of ostriches and emus with respect to convergent evolution demonstrated a more diverse paraphyletic characteristic of each species' development. Specifically, emus were found to have significantly slower wing growth rates compared to ostriches, whose development patterns were faster with chickens and tinamous according to their embryonic datasets. Thus, the presence of convergence within flightless birds was now viewed as an accurate representation of their phylogenetic roots.